Good afternoon, everybody. It is slightly afternoon, so it's a pleasure to welcome Dr. Peter Barker here today. Dr. Barker is currently a professor of radiology at Johns Hopkins University. He attended Oxford University, where he received a BA in chemistry and then a Doctor of Philosophy degree in physical chemistry. He did postdoctoral work at California Institute of Technology, and in 1989 he joined the NMR Research Division at Johns Hopkins, and then in 94 he accepted a position on the bioscientific staff in the Department of Neurology at Henry Ford Hospital in Detroit, while also teaching as an adjunct associate professor of medical physics at Oakland University in Rochester. He returned to Hopkins in 1997, where he has remained since, and he's a very renowned researcher in the field of in vivo MR spectroscopy. He's the principal author or co-author of over 200 publications, and we really couldn't get a finer expert to talk to us about in vivo spectroscopy than Dr. Barker. So Dr. Barker.
Okay. Thank you very much. Thank you, Mark, for that very kind introduction. And also I'd like to thank Dr. Halbauer for also inviting me to come here today. And I hope we continue our collaboration that we started when you were at Johns Hopkins, and hope we'll be able to keep it going now you're here in Colorado. So what I'm going to talk about today are some recent developments in MR spectroscopy of the human brain. And I wasn't quite sure who my audience would be, so I have some sort of material at the beginning which is sort of more of an introduction to spectroscopy and talking a little bit how it's used in the clinic today, and then the latter part of my talk is more sort of focused on the sort of neuroscience and research applications. Now, spectroscopy is an old technique. These are the founding four doctors of the Johns Hopkins Hospital. From left to right, Dr. Welch was the first dean of the School of Medicine, was a pathologist. Then there's Dr. Halsted, who was the first chief of surgery. Some of you may know a recent book came out on Halsted, and he was addicted to cocaine and later morphine throughout his whole of his surgical career, interestingly enough. Then there's Dr. Osler, chief of medicine, and then Dr. Kelly, a surgeon at OB/GYN physician. This famous painting done in 1906 by John Singer Sargent. Now, one of the things that people asked is there's a book on the table here in front of them. And what was the book that they were all reading? And it actually turned out it was a text on clinical MR spectroscopy. No, that's a lie, actually. But we didn't have MR spectroscopy in 1906, but actually this is one of the early papers on clinical MR spectroscopy, and you can see it was published in 1989 on spectroscopy of human brain tumors. So this is now 24 years ago, which in the sort of research, I mean, that's a long, 20 years is a long time. Nevertheless, I hope I can convince you that this is still a field where there's active developments and new things going on and being discovered. And I just point out, I'll talk a little bit more about this in a moment, but the very different spectrum you get from astrocytoma compared to normal brain tissue. So the NAA signal in normal brain is gone, and there's this very high choline signal, which is sort of typical characteristics of a brain tumor. So if you look at a normal brain, there are three signals that you can detect. This is a long echo time spectrum, and the largest of which is this one at 2 ppm on the frequency scale, which is from N-acetyl aspartate, which is pretty good evidence that NAA is localized just to neurons, axons, and dendrites. So this is often called a neuronal marker. And it's certainly is almost uniformly observed to go down anytime you have some disease that has neuronal loss or neuronal dysfunction. This NAA peak nearly always goes down. There's another peak from creatine on 3 ppm on the frequency scale, which actually comes from 2 different compounds, creatine and phosphocreatine, which are energy metabolites. And then there's a third peak from choline, which again actually is several different choline-containing compounds. We just call it choline for short, particularly glycerophosphcholine and phosphocholine. And these are compounds that change in a lot of diseases. Glial cells have a lot of choline in them, so that's why you see choline go up in glial brain tumors. But it can also change under other pathologies as well. For instance, when you have active demyelination, the choline is like a breakdown product of myelin, so you can see the choline increase with active demyelination also. And then you don't see it in normal brain, but you do see it when there's some pathology, and that's this lactate signal at about 1.3 ppm, which is this characteristic doublet pattern. This is actually a patient who is having who's having a right MCA stroke. You don't yet see it on the conventional MRI scan, but this part of the brain is ischemic, and ischemia causes the lactate concentration to be increased, and then you can detect it. And you can also see increased lactate in other conditions as well, and I'm going to come back to talk about lactate more later. Now, when you go to short echo times, that spectrum I just showed you was a long echo time spectrum. At short echo times, you get additional signals that you don't see in the long echo time spectrum. So you get signals from glutamate and glutamine, just to the left of the NAA peak here. You also get a signal from myoinositol, which is also present in glial cells in high quantities. And you can also pick up lipid signals as well. Sometimes you get surprises, or peaks you don't expect to see. This is a normal volunteer scanned in the afternoon after they've had lunch. And you see there's this extra peak down here at around 1.2 ppm. It's a triplet. This is actually ethanol. And actually you can get quite a strong ethanol signal in the brain after a fairly modest amount of alcohol consumed. Basically anything which freely crosses the blood-brain barrier in large enough quantities has the potential to be detectable by MR spectroscopy. Now, I want to talk a little bit about how we do MR spectroscopy. And basically you have to define the region of interest in the brain that you're getting the spectrum from. And there's 2 sort of different ways of doing that. There's so-called single voxel techniques where you just pick one region of the brain and you get that one spectrum. And there's a couple of different commonly used pulse sequences that are used to do that. One's called PRESS and one is called STEAM. They're kind of similar in that they both have slice selective pulses applied in 3 different directions, and you basically set up the scanner so that you only collect signal from the region in space where those 3 slices intersect one another. These days, most people prefer to use PRESS because it gives you a little bit higher signal to noise, but you can also use STEAM. I mean, either is fine, really. And then examples of how to use this. Well, one protocol that we do quite often at Johns Hopkins is when we have a patient with suspected temporal lobe epilepsy, and the question is, which of the mesial temporal lobes, the left or the right, is abnormal? And these might be patients who would be candidates for surgery to stop the seizures. And here's a patient who has nocturnal seizures. You can see in the spectrum from the hippocampus on the left, there's a lower NAA signal than on the right. And that can be quite useful information to lateralize the onset of seizures to the left hemisphere. I'd have to say that there's probably only a minority of patients where this is useful in the sense that you already don't know the answer from conventional MRI, but occasionally it can be helpful. Now, the other class of localization sequence, so-called multivoxel sequences or spectroscopic imaging, also sometimes called chemical shift imaging or CSI. And basically the idea is that we start out by exciting a large volume of brain tissue, still using the PRESS sequence, but now instead of a small voxel, we have a large chunk of brain tissue that we're exciting. And then we apply phasing coding gradients, just like you do in MRI, and you basically dice up that large voxel into a whole bunch of smaller voxels, and you get a spectrum simultaneously from every one of those small voxels in the grid there. And again, a clinical example is, again, a patient with seizures and looking at the temporal lobe, and here you can clearly see the abnormal left hippocampus already on the T2 weighted MRI scan. But when you do multivoxel spectroscopy and look at these two voxels, you can see now that like, the choline is up and the NAA is down. There's also a bit of lactate. So this is a more typical pattern for a tumor or perhaps a viral encephalitis, not mesial temporal sclerosis, which might be the other pathology which is being considered. And you can also reconstruct these low-resolution metabolic images from this type of multivoxel data set. So you can make an image of the distribution of choline in the brain or the distribution of NAA. And even though these are low-resolution images, you can clearly see the increased choline and the decreased NAA in the left mesial temporal lobe. Now, a pulse sequence that I am particularly fond of, and have been using this for about 20 years now, is this multi-slice MRSI sequence, slightly different from the press MRSI, which I showed you previously, and that now we're basically exciting whole slices, and we can interleave multiple slices together to get more brain coverage. And there's a few things you have to do. So we have to do some water suppression at the beginning because the water signal in the brain is so enormous, so you have to do a water suppression pulse to get rid of that so you can see the low concentration metabolites. And then you have to do, in this sequence, you have to do this outer volume suppression. You see these red outer volume suppression pulses here, which are applied around the brain. And the idea here is we're suppressing the lipid signals in the scalp, which if we don't do that, we have all sorts of problem with lipid contamination in our spectra, because again, these are very strong signals from the lipid in the scalp unless you suppress them. So using this pulse sequence, so here you can make basically whole slice axial images. This is the brain of a normal adult. And you can see there are some slight regional variations. So for instance, like the choline signal looks higher in the white matter than in the gray matter. It's also somewhat higher frontally compared to the posterior region. And you can pull out spectra from all of these different regions. So like the corpus callosum, the genu and the splenium, and again, notice the genu and the splenium is somewhat different spectral pattern. And then not all gray matter is the same. Actually, there's sort of this frontal region here in the cingulate gyrus, mesial frontal region. Actually, the choline is quite a bit higher than in, say, the parietal gray matter here, where you see it's lower. So these are all normal regional variations that if you're looking at spectra from a patient, obviously you need to know what these normal regional variations are in order to know what's normal and what is abnormal. Now, I just want to talk a little bit about using this pulse sequence in brain tumors. As we've already seen from my second slide, that the characteristic of pattern of a non-necrotic high-grade brain tumor is that it has a very high choline signal. So here's GBM. Here's the spectrum here, showing very high choline, reduced NAA, compared to the contralateral hemisphere, which has a normal pattern. And you can, one of the things which is interesting here is if you look at the choline image, you can see that this high focus of choline is only in the posterior aspect of the lesion. And we believe what this means is in this case is this is where the high concentration of the tumor cells is. And on the more anterior part of the lesion that you see on the regular MRI scan, this may be more edematous than tumor. And so if you're going to target a biopsy or target treatment, tell the neurosurgeon to focus more on the posterior aspect of the lesion where the highest density of tumor cells may be. Now, do all brain tumors have a high choline signal? Well, actually, no, they don't. So here is an example of a low-grade brain tumor, a grade 2 oligodendroglioma. Now, when we look at the spectrum from the lesion, actually the choline signal is about the same as in the contralateral hemisphere. But clearly the NAA signal is much reduced compared to the contralateral hemisphere. So just, this is one study, actually from my colleague Alberto Bizzi in Italy. This is a study of 58 patients. He's looking at the choline level as a function of the grade of the brain tumor, either mixed oligoastrocytomas or pure astrocytomas going from grade 2 to grade 3 to grade 4. And the choline signal actually measured different ways, either normalized to the contralateral hemisphere or expressed as a ratio of choline to Na or choline to creatine. What you can see is there is a trend for the choline to increase as you go up to higher grade brain tumor. But I also draw your attention to the error bars on all of these charts, which shows there's a lot of variability in this data. So you can't measure a choline signal and say with confidence, yes, this is grade 2 or grade 3 or even grade 4. But it can help you. There is a general trend for increasing choline with increasing grade of tumor. We actually asked a slightly different study in this published, which came out about 4 years ago. We weren't trying to grade the brain tumors, but we were actually asking a different question. Is when a patient presents with a new brain lesion, not yet biopsied, is it a malignant lesion or is it a benign lesion? And this is important because if you truly believe it to be a benign lesion, you probably don't want to biopsy it, because obviously a biopsy is an invasive procedure which has its own risks and so on. So if you are very confident that based on the imaging characteristics it's a benign lesion, you can say hold off on the biopsy and just follow the lesion over time to make sure it's not getting bigger. So these are actually the results of the study, and this was using MR spectroscopic imaging and also perfusion imaging. And I forget the number of cases now. I think it was about 50 cases. We basically constructed ROC curves, which are a measure of the accuracy of a diagnostic technique, for perfusion imaging, for spectroscopic imaging, and the two combined. So in an ROC curve, you plot the sensitivity versus 1 minus the specificity. If you have a perfect test, an ROC curve will go straight up the y-axis and then straight across like this, and the area under the curve would be 1. So that would be basically perfect classification. And you can see for perfusion, we actually got an area under the curve of 0.86, and for MRSI 0.92. If you put the two together, it's 0.96. So actually very good sensitivity and specificity for distinguishing malignant from benign lesions. And just to show you a couple of cases, so here's a primary CNS lymphoma. It's a high-grade aggressive brain tumor, so you can see the high choline signal in the lesion down here. You can actually also see on the perfusion image, looking at the blood volume, the CBV, or increased CBV in that region. And then an example of a non-neoplastic lesion, a patient with meningoencephalitis, actually sort of similar appearance on conventional MRI scans. But now actually when you look at the metabolic images from spectroscopy, you can see low choline, actually low creatine and low NAA. Everything is low. So a completely different pattern. So this really helps in diagnosing this as a non-neoplastic lesion. Now, I don't want to give you the impression that it works every time. Here's one case, which is actually a pitfall. This turned out to be a tumor factor MS lesion. And at first sight, you would think this was an astrocytoma because it's got a high choline signal. Here, it's got a little bit of lactate, Na is low. Looks just like a brain tumor. But there was actually a couple of things in this patient that convinced us that this was not a tumor. And the first of all was actually on the perfusion scan, the low blood volume. And then there's also this incomplete rim pattern of enhancement, which is much more characteristic of a tumor factor lesion. So this patient was actually scheduled for biopsy, but it was decided not to biopsy this patient, and they were treated with steroids. And you can see 3 weeks later, this lesion has almost disappeared altogether. So we're very happy not to biopsy or operate on this patient. While I'm talking about brain tumors, one other thing I want to mention, and that is looking at treatment effects. So typically, patients with high-grade brain tumors, they're going to get surgery, they're going to get chemotherapy, they're going to get radiation. Actually, with temozolomide, there's a slight increase in the survival compared to before temozolomide was available. And there's a lot of interest now in anti-angiogenic agents for treating brain tumors also. One of the problems is when you're looking at these patients on follow-up with conventional MRI, it's very difficult to know if you're seeing a treatment effect or if you're seeing regrowth of the tumor, because they can both have very similar appearance on conventional MRI. So I think actually one area where spectroscopy is very useful is, this is actually the original paper. It's hard to find. It's from the Canadian Journal of Neurological Science. But I did manage to dig up a copy of it and made this slide. And you can see this patient, this is a patient with pathology proven. Now, this patient actually had both recurrent tumor and necrosis in different parts of their lesion. And you can see in the necrotic region here how in necrosis there's low levels of all of the different metabolites, whereas in the area of tumor regrowth you can see the high choline signal. And I think this is actually very promising methodology for distinguishing tumor regrowth from radiation necrosis. A more systematic study done somewhat later by my colleagues in Detroit at Henry Ford Hospital showed differences in the choline signal, higher in tumor, lower in necrosis, which is statistically significant, as well as higher lipids in necrotic tissue compared to recurrent tumor. But also highlight one problem with this, in that some of the lesions had mixed pathology, so both necrosis and tumor regrowth in the same region. Spectra from those regions weren't statistically significantly different from either pure tumor or pure necrosis. So basically, if you have mixed pathology, it's basically with spectroscopy, you can't really make any comment on about it. But if you have pure necrosis or pure tumor, then the spectroscopy may be helpful. Now, I also want to talk a little bit about hypoxia and ischemia, because I know this is something that Dr. Halvar is very interested in. And so, while perhaps this isn't something that's used that much clinically, spectroscopy actually still, I think, it can be quite informative. So just as a little bit of background to start with. So this is a very old study from 1987. I think this was done in mice or gerbils or some small animal, but not a human study, obviously. But basically, yes, this is the gerbil. In a gerbil, if you occlude the carotid arteries, you can produce global ischemia because they don't have posterior circulation. They don't have a circular willis. So it's actually a convenient model for studying global ischemia. Basically, once you have insufficient oxygen supply to the brain, glycolysis, the metabolism of glucose, switches from oxidative pathway to an anaerobic pathway, which ends up with the synthesis of lactate. So basically, in this study, what they did, they slowly reduced the cerebral blood flow. So normal values around sort of 40 to 60 range. And then they tied off one or both carotid arteries. And they measured the lactate level relative to NAA. You can basically see once you drop the blood flow below about 20 mLs per 100 grams per minute, you start getting synthesis of lactate, and it actually shoots up to quite high levels very, very rapidly. So this is basically, this is believed to be a flow threshold below which you start getting lactate synthesis. We did some old studies, again, this is like back in the 90s, basically occluding the middle cerebral artery in a baboon model. So this is a model of focal ischemia, it's not global ischemia. But it was permanent focal ischemia, the artery stayed occluded. And this following these animals over time. So actually, within less than 20 minutes, about 15 minutes into the occlusion, you can already see a small lactate signal coming up. And then just progressively over time, presumably as the infarct got larger and as more lactate was being synthesized, the lactate progressively built up. And then concomitant with that, there was actually a slow decrease in the NAA signal, presumably reflecting the neurons dying. And there's actually been various papers on this, looking at the time course of NAA decreases and lactate increases in focal ischemia. And at one time I thought this might be potentially useful for guiding stroke therapy when you have an acute stroke patient. doing spectroscopy to see what the state of the neurons was and whether you should attempt to reperfuse or not. But I have come to the conclusion I think it's too complicated to do a spectroscopy study in an acute stroke patient, and this information is actually available from other things like diffusion and perfusion imaging. Nevertheless, just showing you a few examples. So this is actually the patient I showed you at the beginning with the right MCA occlusion. This is actually about 24 hours after the stroke onset. And you can see in the lactate image here the increased lactate in the right MCA distribution. And followed up 1 week later, interestingly, so the patient had developed an infarct in the right basal ganglia where the NAA is down and the lactate is up, so typical of an infarct. But the lateral region, which if I just go back for a second, you see this lactate here in the cortical regions, has basically disappeared. And this patient actually reperfused and actually improved clinically a lot. And this kind of explains why, because those functional cortical regions are now no longer ischemic, so they're functioning again. However, this patient subsequently deteriorated again, and at 5 months had basically gone back to being hemiplegic again. And at 5 months, there's really no lactate, but we can see some chronic changes now. So there's more widespread reduction of NAA in the right hemisphere. Interestingly, the choline is also up somewhat in the right hemisphere as well. And even though actually the infarct is kind of small, this is a sort of slit-like infarct on the regular MRI, probably a lot of this actually right hemisphere is abnormal, and that kind of explains why the patient's symptoms have returned. So this sort of summarizes the sort of time course of changes that you see in the brain as a function of time post the onset of ischemia. So in the early stages, you have increase of lactate, decreasing NAA, and in a sort of more chronic situation that you sort of have low NAA and choline up, perhaps due to gliosis. You may see some lipids from membrane breakdown products, and you may actually see a little bit of lactate in the chronic stage. And it's not clear if this is ongoing ischemia or it might be from macrophage metabolism. So basically what you see depends on the stage of the stroke. I also just want to mention the other perhaps area of application of spectroscopy in terms of ischemic damages in neonates and evaluating birth asphyxia. And this is a newborn scanned 16 hours after a difficult delivery. If you look at the conventional MRI scan, well, these are very difficult to interpret. You have to be a real expert specialized in this to be able to say whether that's normal or abnormal for a 16-hour baby. It actually is considered normal. But you can already see on the diffusion-weighted imaging, there's some restricted diffusion here in the thalamus, and just a tiny little lactate signal in the thalamus. This would be, I think, quite hard to call as abnormal because it's so small. But four days later, the restricted diffusion has increased, and there's now very clearly a lactate signal. And at this stage, there are some subtle abnormal signs in the T1 weighted image. So the posterior part of the internal capsule should be bright, and it's not in this image. And then continuing by 8 days, more abnormalities, again restricted diffusion, although in a different pattern, and also still some lactate. So I also just want to point out that interpreting spectra from newborns is also kind of difficult because the normal spectrum of a newborn is quite different from an adult. So here at 4-day-old in either white matter or gray matter, see the NAA is pretty low compared to either a 4-year-old or adult brain. And also the myonostal and the choline are high. And they change pretty rapidly, particularly over the first 2 years of life. You can see at 5 months, NAA coming up, choline and myonostal going down. And by about 4 years of age, at least in these two posterior brain regions, the spectra look similar to what you would get in an adult. But it's actually pretty difficult, if you're looking at, say, a 6-month-old child, say, with developmental delay or something like that. You really need to know very accurately the normal age-related variations to say what's normal and what's abnormal. Finally, just one thing to watch out for as well is that I showed you an ethanol signal earlier. Well, here's another exogenous compound. Here's a neonate with hypoxic ischemic encephalopathy. Scanned at 26 hours. There's a very large lactate signal, so that's clearly abnormal, it's ischemic damage. But right next to it, there's another signal, another doublet. Looks kind of just like lactate, but it's shifted over a little bit. This is from propanediol, which is the drug delivery agent given when you give phenobarbital to stop neonatal seizures. It's usually given, this is the vehicle, and this also gets into the brain in large quantities. So you have to watch out that if you have a neonate and you see a signal in this region of the spectrum, you've got to measure its chemical shift to make sure it's, if it's lactate, it's 1.3 ppm, but if it's propanediol, it's about 1.1. Now, what happens if you have sort of more subtle ischemia or hypoxia? So this is actually an example of a patient with a high-grade carotid stenosis. And you can actually see, so it's on the left, on the left-hand side, spectra 1, 2, 3 here. There actually is a lactate signal, which you don't see in the contralateral hemisphere, also lower NAA. Basically, probably what's going on here is that this is a patient who is really at the borderline of becoming ischemic and having a stroke. There's probably just enough blood flow, getting to the left hemisphere to keep the hemisphere functioning. But at the same time, there is a small amount of ischemia, and that's probably what's causing this increase in the lactate signal, just a small increase in lactate. And if you reperfuse these patients by doing endarterectomy, you typically, the lactate goes away, but actually the NAA signal doesn't seem to recover, at least in this publication from the Dutch group about 10 years ago. That's what they found. And even more subtle hypoxic injury, if you like. If you just basically reduce the amount of oxygen that you're breathing, so this is an experiment with like graded hypoxia. This is done in humans. It's safe to do this in humans. So normally we breathe about 20% oxygen, but if you decrease the inspired oxygen level to about 12%, so this is probably like going a little bit higher than Pike's Peak, maybe somewhere in the Himalayas, you'd be like breathing this amount of oxygen. Actually, the lactate signal does go up in the brain. Now, earlier I told you that you can't measure lactate in the normal brain, and that's sort of true, but not entirely true. Is if you use sophisticated enough techniques, you can actually quite accurately measure lactate in the totally normally oxygenated brain. And you have to use what's called a spectral editing technique, this mega press technique. I'm going to talk more about that in a moment. And looking at large volume of tissue, you can actually get a quite robust lactate signal. So in this subject who's breathing 12% oxygen, you can see the lactate. This is actually a pretty big increase. It's about a 90% increase in the brain lactate signal. Compared to normoxia. And actually how you interpret this is a little bit tricky, because it's possible that some of this lactate may be being synthesized in the body and then just carried into the brain through, obviously through the blood supply of the brain. But nevertheless, it does seem that there is an increased lactate signal in the brain that could be used to study hypoxia, very mild hypoxia. Now, I want to shift gears a little bit and now talk about other molecules and neuroscience. And a molecule that's really more and more interested in over the last, well, at least 10 years or so is GABA, gamma-aminobutyric acid, which is the most abundant inhibitory neurotransmitter in the brain. And, GABA is involved in many, many different aspects of brain function, including brain development, epilepsy, obviously it's very important, but also sleep, alcohol, drug addiction. GABA is important, as well as in psychiatric diseases such as schizophrenia. And there are many drug therapies out there which are actually targeted at either changing GABA levels or in the brain or altering the GABAergic system. So clearly, if you could measure this non-invasively with MR spectroscopy, it could be a very useful thing. So here's the problem. So this is actually a fairly recent spectrum from our 7 Tesla MRI scanner. Remember, typically conventional MRI scanners are 1.5 or 3 Tesla, so this is a stronger magnetic field strength. And with the stronger magnetic field, you can actually get very good quality spectrum. So here's the conventional spectrum, with NAA, creatine, choline, and so on, as we saw before. And if we want to look at some of the other molecules, so in red here, this is the spectrum from glutamate in this spectrum. So you can see glutamate actually can be pretty well measured. Also glutamine can be, actually fairly well resolved from glutamate in this spectrum. But the blue spectrum, which I'm not sure how well you can see it, but there's one peak here at 1.9, another one here at 2.2, and then there's another peak at 3 ppm. That is the GABA signal. And you can see how much smaller it is than these other signals. And it's very hard to measure it from a spectrum of this type because, one, it's a small signal, and two, it's totally overlapped with these other larger resonances. So the solution to this, as I just talked about with lactate, is to use an editing sequence to selectively detect the lactate signal, get rid of all those other compounds that you don't want. And this was actually first published by Doug Rothman, actually quite a long time ago in 1993. And basically the idea is you do 2 scans. You do one scan with the echo time 1 over the coupling constant between these 3 and 4 protons here. And then you do a second scan where you actually apply these editing pulses, these frequency selective editing pulses, to the C3 protons, which are at 1.9 ppm. And basically what that does is that changes the pattern of this multiplet of the C4 protons. So here the outer lines are negative, here they're positive. So if you subtract those 2 spectra, you basically get this sort of pseudo-doublet, as it's called, which is the GABA signal, and then everything else in the spectrum, so long as it's not affected by these editing pulses, will subtract out. So you end up with hopefully a pure GABA spectrum. So here it is, an example of a 3 Tesla scanner in practice. So GABA is a low concentration molecule, so we have to sample quite large volumes of brain tissue. This is 27 milliliters. And the editing pulse applied here at 1.9 ppm, and then here is the GABA signal around 3 ppm. Typically in vivo, you don't actually see that doublet pattern. It's more of a sort of just a broad hump like this. And the way these experiments are done, actually you also get some co-editing signal from glutamate and glutamine over here, but it's at a different frequency from GABA, so it doesn't really interfere with the GABA measurement. And my colleague at Johns Hopkins, Richard Eden, has been doing a lot of work on GABA over the last few years, in particular in applications in brain function. And I'm just going to show one slide from one of his many studies. And this is a study of normal subjects. And first of all, they have an fMRI scan, visual stimulation. So here you see the activation in the visual cortex. And what you can see is in participant 3 here, they have a much larger area of activation than in participant 2. And this is normal. When you do fMRI, different people have somewhat different amounts of activation, even in normal subjects. And basically what he did is he correlated this bold signal change with their resting brain GABA level in the occipital cortex. So you can see in participant 2, this is the one with less activation, they've actually got a larger GABA signal than in participant 3. So this kind of makes sense, right? So the more GABA you have, remember GABA is an inhibitory neurotransmitter in adults. More GABA, you have less activation. And you could say, well, this is just one study and there's a small number of data points and so on. But actually this type of relationship is holding up now in many, many different activation paradigms and different studies. So this actually could be an important sort of cofactor when analyzing fMRI data, is also to know what the brain GABA levels are in the subjects undergoing the activation study. Also, we're working quite hard now on looking at GABA in psychiatric disease, and this is just some fairly recent results looking at GABA in 2 different brain regions in patients with schizophrenia, either the anterior cingulate or the centrum semiovale white matter. And we found actually there was lower GABA in the older schizophrenia patients who also have longer disease duration than younger patients with schizophrenia. And we're not quite sure what this means. It might be a medication effect, or it might actually be something that the disease is causing. So we still need more studies to understand it. But it seems like it does have some functional significance. So for instance, looking at the anterior cingulate GABA, getting a nice correlation with this task of attention coding, positive correlation. So the more GABA you have, it seems like the better your attention is, which again maybe kind of makes sense, that you get less easily distracted by other things. And then finally, I just want to mention that nearly all of the GABA studies you'll see done in the literature right now are done using single voxel spectroscopy, this megapress technique, where you basically pick one region of the brain and you get the GABA signal from that region. But it is possible to actually do spectroscopic imaging of GABA, so basically map out the GABA distribution in the brain. And this is our sort of first attempt towards doing this, where we sort of started out with our regular, just like that multi-slice pulse sequence I showed you back at the beginning. We basically start out with the same pulse sequence, and then we add into it the editing pulses to select for GABA. So you do basically 2 MRSI scans with and without the editing pulses applied, and you subtract the 2 of them. And then this is our first attempt at this, and I think we're going to, as time goes by, it's going to get better. But actually already quite happy with this. So you can see these beautiful high-resolution spectra of GABA. And as expected, gray matter having higher GABA levels than in white matter. And this is probably a little artifact up here in the right frontal region. But I think in the future, particularly as we go to higher field MR systems and using more sensitive RF coils, that this GABA spectroscopic imaging may become important both for neuroscience and clinical applications. Now, the last part of my talk, I want to talk about going to higher magnetic field strengths. So, as I mentioned, most MR spectroscopy clinically is done at 1.5 or 3 Tesla. And that's interesting. Okay, good. You still have a display. I think I lost my display down here. But there are various advantages and disadvantages to going to higher field strengths, such as 7 Tesla. So from a technical viewpoint, the advantages are that you have higher signal-to-noise ratio, expected to go pretty much linearly as the function of the RF field strength. And you also have increased chemical shift dispersion. So basically what I mean by that is that the peaks in the spectrum get better resolved or pulled further apart. And I'll show you that in a minute. So you can measure the different compounds more accurately. But there are also a number of technical factors that I'm not really going to go through in any detail, but there are actually also some disadvantages to higher field strengths, which are sort of mainly engineering challenges, really. But some of them are a bit more fundamental, things like magnetic susceptibility effects, increase linearly with field strength. There's really not much you can do about that. Relaxation times change also. And something I didn't put on this slide is also cost. These days we're all concerned about how much everything costs. And of course, the 7 Tesla magnet is, they say it's about $1 million a Tesla. So the cost of the 7 Tesla scanner is much, much higher than the cost of a 3 Tesla scanner also. So here are examples of spectra, more or less recorded under the same conditions, going from 1.5 to 3 to 7 Tesla. And you can clearly, I hope, appreciate the increased spectral resolution. So whereas at 1.5 Tesla, it's very hard to separate glutamate or glutamine. At 7 Tesla, they're quite well resolved, and you can start seeing other peaks too, glutathione, silinositol, taurine, all things that you really have a hard time measuring from the 1.5 or the 3 Tesla spectrum. So I was really curious, this is back in 2005, I was really curious to see if we could do spectroscopic imaging on the 7 Tesla system, because at that time nobody had done it. We didn't yet have our own 7 Tesla scanner at Hopkins, but we were collaborating with Philips, and we went to the Philips factory in Cleveland where there was a 7 Tesla scanner. So we just went for the day, and we put a volunteer in the magnet, and we just ran our standard 3 Tesla spectroscopic imaging protocol. And to my surprise, it actually worked, And so we got these beautiful high-resolution brain spectra. And I actually draw your attention to this little bump on the side of the NAA peak. It's actually well known that this is coming from N-acetyl-aspartyl glutamate, or NAAG, which is a neurotransmitter. And I was really amazed to see this, because I'd never seen that before at 1.5 or 3 Tesla. So I felt this was an opportunity to measure a new compound that we couldn't do before. And so I was basically able to make metabolic images of choline and creatine and NAA, just like I do at 3 Tesla, but then I could also make glutamate and myoinositol images that I wasn't able to do previously at 3 Tesla. So then we kind of waited, and then in end of 2008, we got our own 7 Tesla magnet. Here you can see it being delivered on the crane. So we basically, since we've had this system, we've been trying to figure out what it's good for and what it's not good for. And actually, one of the things it's very good for is anatomical MRI scans. Basically, just using the increased signal to noise of 7 Tesla compared to lower field strength, so you can go down to smaller voxel sizes or higher spatial resolution. So these are isotropic 3D T1 weighted images done with a 0.6 millimeter spatial resolution. And I think I have a movie. Let's see if the movie runs. Yep, here it is. Yeah. So you can see this lovely gray-white contrast in these images. And you really can't do this at 3 Tesla. At 3 Tesla, you can get this sort of image with about a 1 millimeter spatial resolution. So this is about 8 times better in terms of the voxel size. So when you go from 1 to 0.6 and then you cube it, it's about 6 to 8 times higher spatial resolution. So I think these are going to be, I don't have any evidence for it yet, but I think these sort of images are going to be very useful for looking at things like, subtle abnormalities, cortical malformations, or looking at the hippocampus in detail in patients with epilepsy. But probably many other applications too, looking at cortical thickness and so on. So it's good at anatomical imaging. It's also good at spectroscopy, as I also mentioned. So this is some recent data that we did looking at the anterior cingulate, quite a large voxel size, because we were actually interested in the neurotransmitters. But if you look at the conventional spectra, this is one subject scanned 1 week apart. This is visit 1, visit 2. The spectra, this only took about 90 seconds to record these spectra. You can see the good signal to noise and the very similar spectral appearance between the two visits. In fact, when we quantify these spectra, it's a little bit complicated slide, but in blue are the metabolite concentration from the first visit, and green from the second visit, and then the difference is this sort of brown yellow type color. You can see for most metabolites, actually even GABA, interestingly, is actually pretty reliable. But glutamate, glutamine, and when you look at things like NAA, or total NAA, very high degree of reliability in these measurements. So the average coefficient of variation for all metabolites was about 6%. And then the bigger signals in the spectrum, such as the NAA signal, it was more like 2 or 3%. So you can do very accurate spectroscopy measurements also using the 7 Tesla system. I just finish, this is my last slide, by showing some preliminary data. This is actually a study of patients with Huntington's disease, where we're looking at, actually in this case it's the posterior cingulate. And these are patients who are genetically known to be carriers of the Huntington's disease, but they are not yet developed overt symptoms of Huntington's disease. So these are sort of prodromal Huntington's patients. But if you do extensive neuropsychological testing on these subjects, you can tell that they're not normal. And there's this so-called MoCA, the Montreal Cognitive Assessment Score. Interestingly, we found good correlations between NAA and the MoCA score, also glutamate and the MoCA score, kind of indicating that there is subtle neuronal damage in these patients that you can pick up with a spectroscopic measurement, before they sort of develop the overt symptoms of the disease. And I think this could actually be very important, because if you're talking about developing a treatment for Huntington's disease, you want to apply it in the early stages of diseases before extensive neurodegeneration has occurred. And obviously, if you're doing a treatment trial like that, the ultimate measure is going to be, how long do you delay the onset of symptoms for, or how, or do you cure the disease? But if you're going to do a treatment trial like that, you have to wait a long time to know the outcome, whereas if you have some imaging biomarkers which can sort of monitor this early phase of the disease accurately, then that could be very useful for accelerating a treatment trial. So also there were group differences between the Huntington's patients and the controls, so the NAA and the glutamate lower in the early HD patients compared to the controls. Well, I think I've talked long enough, and I've covered a lot of different ground in a short period of time. I hope it wasn't too confusing or bewildering. I want to thank my colleagues at Johns Hopkins who have done much of this work, contributed to the data that I showed today, as well as our funding from the NIH and also support from Philips Healthcare. And I thank you for your attention, and I'm happy to answer any questions you might have. So, thank you. Anybody like to ask a question? Yeah. Mm-hmm.
Are any researchers in, say, Germany or France involved in this new technology?
Well, yeah, MR is really a worldwide effort. Particularly, if you're talking about the high field systems, there's probably about 50 or 60 of them worldwide, of which maybe perhaps 20 or so are in the United States, and the rest is sort of distributed around Europe and Asia. So it's very much it's a worldwide community. So.
Is there more than one supplier of the hardware that's needed?
Yeah, so there's 3 main manufacturers of MRI scanners. GE in the States, then Philips in the Netherlands, and Siemens in Germany. And all 3 of them manufacture high-field MRI scanners. Yeah. So.
I did have a question to GABA.
Mm-hmm.
When you saw 7 Tesla, that the first one was just useful.
Mm-hmm.
but the gathers also come to these peaks. Can you just somehow separate the dispatch together? Does that make sense?
Yeah, that's really a very, very good question, and it's something we're working on actually quite hard right now. Is that so? There's sort of two ways you can measure, GABA and glutathione. You can measure it from like a conventional spectrum, and you're sort of curve fitting these very small peaks. And interestingly, actually, the curve fit, if you look at the reproducibility of the curve fit, is actually pretty good, but we're always a little bit concerned because we wonder if some of that curve fit might be GABA and some of it might be a macromolecule or the baseline. So the other way of doing it is to use a spectral editing sequence. So you can use a spectral editing sequence at 7 Tesla just like you could at 3 Tesla. And you can edit for GABA and you can edit for glutathione. Actually, we have a paper under review right now where we're comparing these two things. I could send you a copy of it if you like. And it's interesting, actually the spectral editing is if you look at the reproducibility, it's not as good as the conventional spectrum. But we think it may be a purer measurement because we know when we're editing, we're pretty sure we're just getting the molecule that we think we're getting, So.
Do you think it's because that if you edit them, you see the low amount metabolized and they do have a greater
I think that the variability we see is more of a technical variability than it's a question which comes up. For instance, we work with some sleep researchers, and we're measuring GABA in that study, and they're very concerned about the time of day that we measure the spectrum at, because their hypothesis is that GABA will change, for instance, as depending on your sleep-wake cycle. But actually, we haven't seen any variation in GABA as a function of that cycle. Generally, we think the variability is coming more from it's technical variability rather than a biological, but not 100% sure about that. So, yeah.
I actually have two questions. There's different technologies. I'm wondering if they could be applied to spectroscopic imaging. One of them is the new dual excitation cultures. Have you put some energy into using that for your editing?
So you're talking about the sort of dual transmit or multiple transmit systems? Yeah. Actually, unfortunately, on our 7 Tesla scanner, we don't have a multi-transmit hardware yet. we hope to get it. But I'm quite sure it's going to really improve things when we get it, because one of the big problems with the 7 Tesla scanners right now is if just a single transmit coil, it's very inhomogeneous transmit field, and that give you all sorts of problems, suboptimal sensitivity, things like that. So I'm quite sure when that becomes available to us, and other places actually already doing it, as you know. So I think that's going to be a big technical advance. Yeah. So. Yeah, so I didn't talk about it today, but we actually have done quite a bit of work also with multiple receiver channels, multi-coil systems, and you're absolutely right. They do improve the signal to noise a lot, particularly in the cortical regions close to the coil. And the other thing you can do with them when you've got multiple receiver channels is you can use parallel imaging techniques like SENSE or SMASH that people are very familiar with MRI. You can do exactly the same thing basically with spectroscopic imaging also. And so you can use that to reduce the scan time. So that multi-slice sequence that I showed you at the beginning, when we first started doing that, this is 20 years ago, on a single channel 1.5D system, that took 30 minutes to collect the data set. Actually, these days, say on a 3T system with a 32-channel coil and sense acceleration, which is very practical, I think. That same data set could be collected in 3 to 5 minutes. So that makes a huge difference in terms of the sort of applicability for clinical studies, obviously. That
Once it gets higher, to be able to create that faster time for more samples, you have to sample that data over and over to be able to really improve your spectrum.
Yeah, so you can sort of trade, if you've got more signal, so you can sort of, you can either, shorten the scan time if you get the same signal to noise, or you could keep the same scan time and go to perhaps a higher resolution, something like that. So, there's a lot of sort of trade-offs you can do as the hardware improves, higher fields or more channels. It definitely improves the quality. And I'm hoping one day I'll have like a 256-channel head coil. That would be nice. Yeah. Yeah, that's a very good question. So yes, so I didn't mention that. So basically, with spectroscopy, we're only looking at small molecules. So if you have like a protein, or some other macromolecule, it basically gives you get very broad lines in the spectrum. You basically can't detect it. So basically, with spectroscopy, all these there's about 17, 20 different compounds that have ever been detected in the brain using spectroscopy. They're all they're probably like sort of, the molecular weight of sort of 200 or less, so they're all small molecules. So, yeah. Yeah, so basically it's to do with when you have a large molecule, it like doesn't tumble, it doesn't move, it moves more slowly, and that actually causes the lines to get broad in the spectrum. So that's kind of a fundamental limitation of spectroscopy. So. Okay, I think that's it. Okay. Yeah, good. Yeah, so I hope people found it interesting.